Hello friends, welcome to biology. I'm Mr. Ryan. Today we're going to be talking about chapter 7.1, Life is Cellular. So, what's the smallest part of any living thing that still counts as being alive? Can we just keep dividing living things into smaller and smaller parts, or is there a point at which what's left is no longer alive? As you can see, there is such a limit. The smallest living unit of any organism is the cell. So if you are a living organism, the smallest unit you are made out of is the cell. It was not until the mid-1600s that scientists began to use a microscope to observe living things. In 1665, Englishman Robert Hooke used an early compound microscope to look at a non-living thin slice of cork. Of course, cork is a plant material. Under the microscope, cork seemed to be made of thousands of tiny empty chambers that Hooke called cells. How did he come up with that term? Because in Hooke's day, there were monasteries, and monks lived in these little rooms called cells, and that's where he came up with it. The term cell is used in biology to this very day. Today, we know that all living things are not empty chambers, but contain huge arrays of working parts, each with its own function. Here's Hooke's drawing of the cork cell, and you can see, okay, that's what he drew. Pretty good drawer. In Holland, Anton van Leeuwenhoek examined pond water and other things, including a sample taken from a human mouth. He drew the organisms he saw in the mouth, which today we call bacteria. So you can see these little circular things, that's bacteria, and these little rod things is bacteria. Soon after Leeuwenhoek, observations made by other scientists made it clear that cells were the basic units of life. In 1838, German botanist Matthias Schleiden concluded that all plants are made of cells. Okay, all plants are made of cells. The next year, German biologist Theodor Schwann stated that all animals were made of cells. And in 1855, German physician Rudolf Virchow concluded that new cells could be produced only from the division of existing cells, confirming a suggestion made by German Lorenz Oken 50 years earlier. These discoveries all are summarized in the cell theory, a fundamental concept of biology and something that you need to know. Number one, all living things are made of cells. Number two, cells are the basic units of structure and function of living things. Three, where do these cells come from? New cells are produced from existing cells. So how do microscopes work? Most microscopes use lenses to magnify the image of an object by focusing light or electrons. We're going to be talking about light microscopes and electron microscopes. Make sure you pay attention to the differences. A typical light microscope allows light to pass through a specimen and uses two lenses to form an image. The first set of lenses, located just above the specimen, produces an enlarged image of the specimen. The second set of lenses magnify this image still further. Because light waves are diffracted or scattered as they pass through matter, light microscopes can produce clear images of objects only to a magnification of about 1,000 times, which I think is pretty good. And here's an example of a light microscope. Here's the eyepiece. Here's the objectives. Here's where you put the specimen. This mirror, we got to get light. It's a light microscope. We got to get light up to the specimen, so we use that mirror. Another problem with light microscopy is that most living cells are nearly transparent, making it difficult to see pictures within them. Using chemical stains or dyes can usually solve this problem. Some of these stains are so specific that they can reveal only compounds or structures within the cell. Some dyes give off light of a particular color when viewed under specific wavelengths of light, a property called fluorescent. Fluorescent dyes can be attached to specific molecules and can then be made visible using a special fluorescent microscope. Fluorescent microscopes make it possible to see and identify the locations of these molecules, and even to watch them move about in a living cell. Electron microscopes reveal details 1,000 times smaller than those visible in the light microscope. So you can see more things with an electron microscope. They can actually see things that are smaller. Electron microscopes can be used to visualize only non-living preserved cells and tissues. So that's very important. When you use an electron microscope, you can only look at non-living and preserved cells and tissue. And here's an electron microscope. As you can see, it's much more complicated than the light microscope that I showed you. But it can get you more detail. Light microscopes can be used to see cells and cell structures as small as 1/1,000,000th of a meter. So 1/1,000,000th of a meter. A meter is a little bit larger than a yardstick. To study something smaller than that, scientists need to use the electron microscopes. Electron microscopes use beams of electrons, not light, that are focused by magnetic fields. Electron microscopes offer much higher resolution, so a clearer picture, than light microscopes. There are two major types of electron microscopes: transmission and scanning. Transmission electron microscopes make it possible to explore cell structures and large protein molecules. Because beams of electrons can only pass through thin samples, cells and tissues must be cut first into ultra-thin slices before they can be examined under a transmission electron microscope. Transmission electron microscopes produce flat, two-dimensional images. And here's an example of that. So these are what you see under a transmission electron microscope. You can see the cells are sliced thin, but you do see the details in them. In scanning electron microscopes, a pencil-like beam of electron is scanned over the surface of a specimen. Because the image is of the surface, specimens viewed under a scanning electron microscope do not have to be cut into those thin slices. Okay? Scanning electron microscopes produce three-dimensional images of the specimen surface. And here's what you see with the scanning electron microscope. Really great pictures, really great detail. Okay? Really great pictures. Like I said, you can see the 3D effect. Because electrons are easily scattered by molecules in the air, samples examined in both types of electron microscopes must be placed in a vacuum in order to be studied. Researchers chemically preserve their samples first and then carefully remove all the water before placing them in a microscope. This means that electron microscopes can only be used to examine, get this, non-living cells and tissues, okay? Non-living cells and tissues. So if you use an electron microscope, what you're looking at is a non-living cell. How are prokaryotes and eukaryotes different? Okay, guys, this is very, very important, okay? This is very important. You need to know this in my class. So from here out in biology, we'll talk about prokaryotic cells and eukaryotic cells. And, of course, on the state test, you're going to have to know the difference between a prokaryotic cell and the eukaryotic cells, okay? Number one, prokaryotic cells do not separate their genetic material within a nucleus. Look at the prefix pro. It sounds like no, okay? It sounds like no. Pro. So, prokaryotic cells do not have a nucleus. In eukaryotic cells, the nucleus separates the genetic material from the rest of the cell. So, eukaryotic cells do have a nucleus. So, let's say pro sounds like no, but you, the prefix you, sounds like do. So they do have a nucleus. Guys, this is very important. Although typical cells range from 5 to 50 micrometers in diameter, the smallest bacteria are only about 0.2 micrometers across, so small that they are difficult to see even under the best light microscopes. However, there are giant amoebas that may be 1,000 micrometers in diameter, large enough to be seen with the unaided eye as a tiny speck in a pond water. Despite their differences, all cells contain the molecule that carries the biological information, DNA. So whether you're a prokaryotic cell or a eukaryotic cell, you have to carry the DNA. If that DNA is floating around in the cytoplasm, you're a prokaryotic cell. If the DNA is in a nucleus, you are a eukaryotic cell. In addition, all cells are surrounded by a thin, flexible barrier called a cell membrane. Cells fall into two broad categories depending on whether they contain a nucleus. The nucleus is a large membrane-enclosed structure that contains the cell's genetic material in the form of DNA, which stands for deoxyribonucleic acid. The nucleus controls many of the cell activities. In fact, you'll see later on as we talk, the cell can be thought of as a little city, and the nucleus can be thought of as City Hall. Eukaryotes are cells that enclose their DNA in the nuclei. Okay, and so here is the eukaryotic cells. You can see the example of them: animal cells, plant cells. There's the nucleus. There's the nucleus. What we found inside that nucleus? The DNA. Let's look at the prokaryotic cell. This is the prokaryotic cell. You don't see a nucleus. The DNA is just floating around in the cytoplasm. So what kind of cells, what kind of eukaryotic cells, what kind of organisms have eukaryotic cells? Well, you have animal cells, you have plant cells, you have fungi, which I know some of you guys out there are fun guys, okay, and protists. Okay, prokaryotic cells are generally smaller than eukaryotic cells. Despite their simplicity, prokaryotes grow, reproduce, and respond to the environment, and some can even move by gliding along surfaces or swimming through liquids. Some of those things sound like the characteristics of life we talked about. The organisms we call bacteria are prokaryotes. So remember that, bacteria are prokaryotes. Eukaryotic cells are generally larger, more complex than prokaryotic cells. Most eukaryotic cells contain dozens of structures and internal membranes. Many eukaryotes are highly specialized. There are many types of eukaryotes: plants, animals, fungi, and organisms known as protists. Well, guys, we've come to the end of our recording. Please do all the assignments you're supposed to do. And remember, have fun and enjoy learning. Thank you.